Today is March 13th, 2018. It's Tuesday, and if you're watching this, you were gone for choir or you were sick, and we are recording this real live time. Say hi, kids. All right, so inferring genotypes, this is the in-class lecture. So careers in biology, geologist is someone who studies and traces the descent of individuals or families, and they help genetic counselors determine whether inheritance patterns are dominant or recessive. So genotypes can largely be resolved through analysis. One thing I want everybody to write down is this. Dominant traits are easier to recognize than recessive traits, or you can take a screenshot. If you don't want to write it down, you can take a picture with your camera, guys. Okay, if you have a phone, take a picture. So when studying pedigrees, it's important to note that if it's a dominant trait, that you're only going to see it in the phenotype. Recessive traits, however, can be hidden, and the only time you can see that is if that person is homozygous recessive. So predicting disorders, this is how we do it. First of all, good records equal future predictions. The larger the pool to study from, the more accurate the prediction. The study of human genetics is difficult. Three main things that make it difficult: time, ethics, and circumstances. And typically ethics and time are the two troubleshooters. Bullet point number 2, the larger the pool to study from, the more accurate the prediction. I had a friend that had the BRCA test. As you guys all know, Angelina Jolie a few years ago opted to have all of her female reproductive organs removed. So she had a double mastectomy, she had her ovaries removed, she had her cervix removed, she had her uterus removed, she had it all removed. She had it removed because she tested positive for the BRCA gene, that early onset and aggressive type of cancer for female reproductive organs. And she didn't want to leave her kids without a mom like she was left. So she opted to have that removed. Just so happens my next door neighbor also tested positive for that same gene about the same time, but her insurance company fought her. Why? Bullet point number 2. The larger the pool to study from, the more accurate the prediction. All they had to go off of was her genetic profile and her mother's. Her mother's sisters all had refused to get the test done because they didn't want to know. And because of that, her insurance company wouldn't pay for her to have that study done. So take a look at this. Considering rare recessive disorders and how they can occur in families after many generations. How is this possible? So basically, I'm giving you the answers to questions, okay? So the allele for a rare recessive disorder can remain hidden in the carrier state for many generations. It's not until a family member with the recessive trait has an offspring with another human who is also a carrier with that recessive trait condition can appear. So name one type of condition that shows dominant inheritance and one that shows recessive inheritance. A dominant is Huntington's disease, achondroplasia. Recessive could be cystic fibrosis, albinism, Tay-Sachs, or galactosemia. You have to know for your test those particular disorders and whether or not they're recessive or dominant. It's on your test, okay? So list the effects, phenotypes of each condition listed above. You need to know that as well. If you can't do that, make yourself some note cards because your test is tomorrow. So complex patterns of inheritance, we're on section 2. So basically everything I've taught you guys from this point back, Mendel, the pea plants, it's accurate, but it's wrong, and I'm going to blow your mind now, okay? There's this thing called complex pattern that does not follow along with what Mendel said at all, okay? So I'm going to show you here in just a minute. It's called incomplete dominance. Complex inheritance pattern in which the heterozygous phenotype is intermediate between those of two homozygous parent organisms. Before you learned a dominant allele will mask or cover up the phenotype of a recessive allele. And in order for that recessive trait to be seen, you had to have 2 recessive alleles, right? Well, now they're saying you can actually have something called incomplete, and you have this in between. So if you take this snapdragon plant that we have right here that's red, and you cross it with white, let's say red is dominant and white is recessive, in this case, you'll get pink. And there's different ways to do it, and I'm going to show you that in class as well tomorrow. Another type of complex inheritance is codominance. Take, for example, sickle cell disease. Sickle cell disease is African descent, and it affects about 9% of that population. It's the inability to properly transport enough oxygen. Figure 5 in page 303 in your book shows an individual who is heterozygous for the trait. It causes changes in hemoglobin, the protein in red blood cells, and causes the cells to change into a C shape. So here you can see a normal cell. We have a normal cell. This one is starting to change. This one's starting to change. This one is in a sickle shape. Even if you have sickle cell disease, you can still lead a normal life. However, when it flares up, it's extremely painful. Malaria, interesting fact. Some areas overlap with sickle cell disease. Scientists discovered those who were heterozygous for the trait, meaning they don't show the trait, but they are carriers, have a high resistance to malaria. And because of this, those people have a lower death rate. So sickle cell disease, although it can be bad, was a benefit. Now that we get into complex dominance, take a look at this Punnett square. Before, you guys were used to just seeing capital R for red and little r for white, right? That's the rule. Well, now you're seeing them used as exponents. So here's where it gets confusing. Depending on the person who's writing out the test or the worksheet or the problem, depending on how they like to do it, it depends on how you'll see it. Sometimes you'll see it written as R for red and W for white, and other times you'll see it written like this as an exponent. If you're like me and getting a lot of stuff in that box jumbled up and it kind of like, I don't know guys, like, it just kind of drives you crazy. It's really hard to see. Do you understand what I'm saying? Okay. You don't have to write it as an exponent. Does everybody understand? So what I would do if it were me, I would get a blank sheet of paper and I would rewrite it down without the exponents there. I would find out my answer. And then if that worksheet or that test wanted it in exponent format, then you can always translate that later. But to do the initial problem, do it with how you know. You don't have to add in a whole bunch of stuff there in the beginning. Make sense? Okay.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. We mean that genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig.
So this is what you've been used to up to this point, okay?
Well, that's a Mendelian trait. What we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers, so I don't know, that's not exactly what we envisioned. Well, in snapdragon genetics, there can be 3 phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here big R, and a white flower, little r.
Okay, so here on this side, so this part can get confusing because you're seeing 2 different things happening at once. The left side of the screen is the Mendelian side, which is what you've been doing all along. This is the part that's changing. This is the incomplete dominant.
You are going to get offspring that are big R, little r.
So they all look the same, right? Here's where it changes.
But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross 2 pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink.
Okay, so if it matches big R, then it's going to be that phenotype, which is red. If it matches little r, it's going to match that phenotype, which is white. But if it has a big and a small together, these don't match, right? So if it's heterozygous, if it's a hybrid, it's going to come out something completely different.
Incomplete dominance is different from codominance. Codominance, like a co-worker, that prefix co should make you think together.
Kind of like Destiny and Erica, they're always together. They're codominant.
Work together, the alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this one.
All right, first hour, the bell rang, so we will continue on tomorrow. Make sure you look at video part 2 for today because we went over pedigree chart, and that is due for you tomorrow as well.